Hello, everybody. My name is Daniela Weiskopf. I'm a research assistant professor at the Jolla Institute for Immunology, and I'm very excited today to have the opportunity to present on our most recent study where we were investigating the immune response to a low-dose mRNA vaccine and if it has any effect if you have pre-existing cross-reactive T cells. So for the last 18 months, I have been spending every day to study how our immune system recognizes COVID-19. And we have found out that after natural infection, you have a pretty stable memory response up to 8 months post symptoms onset. And this is just a summary of this publication. So when you compare the immune response parameters, and we've always measured CD4, CD8, B cells, neutralizing antibodies, and IgG titers. So 1 month after infection, the majority of people has all 5 of these parameters detectable. 6 months, the majority of people still have at least 3 or more, as is indicated here by these blue little man. So we were interested in, okay, this is good news for natural infection. How does this look like after vaccination? And the other question that we were interested in is we early on identified this phenomenon that we do see cross-reactivity or responses against SARS CoV-2 in people where we are very certain have never seen this virus. So about 50% of the people had detectable CD4 responses that reacted against peptides derived from SARS CoV-2, but we knew they never have seen this virus because these were blood samples collected in 2015, 2016, so way before this virus was circulating. Also, we were certain that they have never seen the virus because not only we collected it way before onset of the pandemic, but of course we also tested antibody parameters and made sure they have never seen the virus. So these are the 2 questions that we had going into this study. So how long does infection after vaccination last after we knew that after natural infection, at least 8 months, it has detectable responses? And then what is the influence of these unexposed cross-reactive T cells that we have detected in about half of the people that have never seen the virus? So this was a perfect cohort to analyze these 2 questions. How long does T cell memory last in mRNA-1273 vaccinees? And does this pre-existing T cell immunity influence the outcome of vaccination? And we had access to samples from the phase 1 trial of the Moderna vaccine. But important to point out that this was the low dose. So this was 25 microgram of the mRNA vaccine, which we by now, everything that's given, everybody receives the 100 microgram dose. So we thought this was a perfect cohort because, first of all, how long does T cell memory last in the low dose of this vaccination? And also we thought that it's easier to dissect the differences in the subsequent immune response if there is a lower dose, if we want to dissect if pre-existing immunity has an influence on that. So this is the vaccine cohort and the visits we analyzed. So as I pointed out, we have studied people that have received in phase 1 clinical trials the mRNA-1273 vaccine developed by Moderna and NIH. As I said, importantly, the low dose, 25 microgram. We studied 35 different donors, and I want to point out that they were spanned across all of the different age groups. So we had 15 donors in the 18 to 55 year age bracket, 10 donors in the 56 to 70 age bracket, and then 10 donors older than 70 years. So we thought it was important because we didn't want to receive or find out results for just the young cohort. So we were excited to include donors spanning all ages. So also importantly, this is the visits that we have been studying, as you can see on the right side of the slide. So all of these samples had an enrollment visit. So that means this is a blood sample that has been collected before they received the vaccine. And this is important because, as I pointed out, one major question we had was this pre-existing immunity, does this have an influence of subsequent vaccination? So if you don't have a pre-bleed, then you don't know which one of your donors has pre-existing immunity. We then collected 2 weeks after each of the doses. So they received on visit 5, they received the first dose of the vaccine. 2 weeks later, we collected the sample. 28 days later, they received the second dose. 2 weeks later, we collected a sample. And then because we were interested in the durability of the immune response, we waited all the way to 7 months after first dose or 6 months after second dose, like to collect the blood sample and see, can we still detect all of these parameters? So this is going back to the two main questions we had, and I'm going to start with the first question. So how long does T-cell memory last in mRNA-1273 vaccinees? And as I pointed out, we are always interested in studying like not only the cellular immunity, but also antibodies. And so we have also measured that here. So this is the first slide I'm going to show you. So if you look at the antibody responses, and this is measured on the left panel, spike IgG, specific responses, and on the right side, RBD IgG specific responses. You know, we thought this was important to measure both ways because like some vaccines have only the RBD IgG in it, and also like we know it's a major target of antibodies, but also because this vaccine contains the entirety of the spike protein, we wanted to make sure we measure these both two parameters. So we saw that significant responses, if you compare it to the pre-bleed, at all 3 time points post-vaccination were identified. The peak of response was higher at day 43 compared to day 15. So having a second dose truly has a benefit because it raises your antibody levels even higher. And we saw also that responses were maintained up to the 209 days time point, which is like 7 months after the first dose. And you can see here, if you look at the lowest line, still 100% of people responded, meaning they had detectable antibodies. Either way, we measured it after 7 months of vaccination. So that was encouraging because that means like that everybody still has detectable antibodies, even though they have received a low dose of the vaccine. We were then interested in doing a more functional readout. So this is why we measured neutralizing antibodies. So that does not mean only measure the levels of antibodies, but also how functional are they? Like, can they actually neutralize? So this is the results from there. And you can see that we did not, we have a slightly delayed induction of neutralizing response. So on the first 2 weeks after the first dose, we have about 30% of the donors that have neutralizing antibodies. But after the second dose, 100% of the people have detectable neutralizing antibodies. Again, the peak response was highest at day 43. That is the same what we saw for the levels of antibodies. And also, like almost all donors, 88% have still detectable antibodies 7 months after vaccination. So next, we wanted to go to the T cells because like a lot of these antibody results were already published. So we just wanted to measure like them in our own cohort to make sure that that our results are comparable to what has been shown in the literature. But what was not known yet is like the exact distribution of T cells after vaccination. And this is basically how we measured antigen-specific T cells. I will quickly want to like walk you through so that you can appreciate the different ways we have been thinking of this. So on the left side, it shows peptide pools that we have been using. So of course we use the spike peptide pool that spans the entire spike protein because this is the antigen that is covered in this vaccine. But we also covered the non-spike protein just to make sure, because we know that a lot of the pre-existing immune response is targeted against proteins other than the spike. So we wanted to also like include this measurement in our assay. And then of course we had controls. One was like using antigen pools from a different virus. So we knew that a lot of the majority of people is exposed to cytomegalovirus. So this is a great control if you want to measure antigen-specific cells against another virus. So this has been included. And then of course positive negative control, DHA and DMSO, to make sure that we can qualify our assays. The assays we have been used is 2 different ones. The first one is the AIM assay, and you can see here in the middle panel the markers we have been used to identify these cells. AIM stands for antigen-induced molecules, and it allows you to identify antigen-specific cells based on the upregulation of activation markers. And why this is important is because like that is basically allows you to identify antigen-specific cells, irrespective of their cytokine production. So we started using this assay early on because in the beginning it wasn't very clear what cytokines this virus induces. So we wanted to have an unbiased assay that allows the detection of all of the antigen-specific cells. Also importantly, this assay will detect, for example, cells that do not traditionally make a lot of cytokines in the periphery, so such as TFH cells, T follicular helper cells. So this assay allows you to get the entirety of the antigen-specific cells, regardless of what profile, the cytokine profile they induce. However, just like we wanted to have like a level of antibodies, we also wanted to read the functional antibodies, as I just shown you. Same here, we wanted to see the level of antigen-specific T cells, but also their function. So we did actually also run in parallel a traditional ICS assay, which is intracellular cytokine staining. That means we can identify which one of the antigen-specific cells make which ones of the cytokines or combination thereof. These are the cytokines that have been, the cytokine panel that has been utilized in our assay. And you can see here, it's a pretty broad range. It spans from Th1 cytokines, such as interferon gamma, TNF alpha, also Th2 cytokines, as interleukin 4, and then also other cytokines associated with either T regulatory cells, or other substantive helper cells, such as interleukin 17 or interleukin 10. We have a cytotoxic molecule, which is granzyme B, just to see if there's another capability of these cells to have cytolytic responses. So why this is important to cast a broad net of cytokines is because also in the beginning it wasn't very clear what cytokine profile these virus-specific cells will show. There was like concern that they might be biased to Th2, which people were worried about would like more interfere with like pathology. So this is why we have a broad range, and this is why we're using these two different assays for our characterization. So if we go right into the results, this is the CD4 T cell responses, in this case measured by AIM, and you can see again a response like significantly higher at all three time points after vaccination. The first dose already induces a CD4 T cell responses, antigen-specific response in 97%, reaches 100% after the second dose, and also is able to maintain a detectable CD4 T cell responses in 97 to almost everybody up to 7 months. So just what we have seen for like natural infection, also here we see like a very durable CD4 T cell response. And it's important to point out that we are very confident when we measure like responses, memory responses 6 to 7 months after infection or vaccination. This is probably a good indicator like of like how the response is going to be maintained. So we do not expect that this response basically falls off a cliff 7 months. So because you can already see it's a pretty like gradual like maintenance of the response. So that's encouraging. And I also showed you that we have included controls, and I just quickly want to touch base on this. So one control we have been using was the CMV-specific responses. You can see here, we do detect CMV-specific responses in the majority of donors, and they are not changed regardless if we get 1 dose, 2 dose of the responses. And that's important because that means the vaccine does not induce, does not influence other antigen-specific cells outside of the proteins that are encoded in this vaccine. And according to this line, of course, also there's no change in the proteins outside of the spike, other proteins from SARS CoV-2, so they're also not changed. So just to show you as a control that the vaccine-induced response is spike-specific. I told you it's important for us to measure also circulating T follicular helper cells. The reason why we think this is important is because this is the subset that does provide help to B cells to make like highly functioning antibodies. So it's important to know if this subset is actually induced by this vaccine. And this is just for the people that are interested in the way we characterize these. So we know which ones are antigen-specific, SARS CoV-2 spike-specific, T follicular helper cells. And also, if you look at the induction of them, we see that more than 2/3 of donors show T follicular helper cells in the periphery after the first dose, and about a similar amount of donors after the second dose. And about 2/3 still have detectable immune response of T follicular helper cells in the periphery 7 months after the first vaccination. So also here, a different subset of CD4 T cells are readily detectable 7 months out. I told you that we are also interested not only on the level of antigen-specific CD4 T cells, but also on the function. So this is shown here. So this is a cytokine assay. As I pointed out in the beginning, you measure like cytokines that are produced by each of these antigen-specific cells. And we could readily detect interferon gamma, TNF alpha, IL-2, and also the cytotoxic molecule granzyme B. And we can see that readily peaking after the second dose, but also like detectable out up to 7 months of the response. If you are studying vaccines, you're always interested on like the multifunctionality, which is basically how many different cytokines a cell can produce. Can it produce like only interferon gamma or more than one? So we were interested in like, is there cells that can produce more than one of these cytokines? Because we know traditionally from other vaccines that these are typically correlated with like higher efficacy of the vaccine and higher response capability of these cells. And sure enough, if you look at the cells that can produce more than one cytokine, which is shown here for each of the individual donors, and then also shown here is a pie chart. So the broader the pie chart divides for like the green, the yellow, or the orange pie. That means more and more multifunctional cells are induced, and you can surely see here in the beginning, in the pre-bleed, the majority of cells has one function. This increases after the first dose, with more cells acquiring a second function, even more so after the second dose. Where like there is quite a substantial fraction of cells detectable that can make 3 or more of these cytokines, and that is maintained fairly stable 7 months out of the infection. As a control, we run the same analysis on cytomegalovirus-specific T cells, and you can see here that they are not influenced by the vaccine at all. So there is a certain amount of multifunctional cells, and they remain exactly the same on every time point measured. So this is everything I've shown you about the antigen-specific CD4 T cells that are induced by the vaccine. What about CD8 T cell responses? So we measured spike-specific CD8 T cell responses, and we've seen them significantly induced at each of the time points, again having the peak of the response after the second dose, just what we've seen for all of the other parameters we have measured so far. And we do see a quite substantial amount of responses maintained all the way to the 7 months out. So 34 donors, if you want to put that in numbers, developed spike-specific CD4 T cells after the first dose. More than half of them were positive for spike-specific CD8 T cells after the second dose, and about 1/3 maintained AIM-specific responses after the 7 months time point. And just like what we did for CD4, we were not only interested in like the detection of like the frequency of these antigen-specific cells, but also the function. So if you now look at if there is cytokines induced by any of these cells, you can see that the cytokines that are induced are for CD8 T cells are interferon gamma and TNF alpha as majority. There are some donors that can induce after the second dose also interleukin-2. And if you think back, I showed you there's a broad panel of cytokines that we've measured, but I've only shown you about a subset that for CD4 and CD8 T cells, and that is because none of the other cytokines has actually been detected. So we never seen IL-4 induced, we never seen IL-17 induced in any of these donors, neither for CD4 or for CD8. So this is why you always only see like a subset of these cytokines here. So again, we asked the question like, okay, can these virus spike-specific vaccine-induced cells induce more than one cytokine? And this is the same analysis that I've just shown you for CD4. And also here we see the acquisition of functions after the first dose, after the second dose, and fairly maintained all the way to day 229 after vaccination. So again, also for CD8 T cells, we saw that the majority of cells does induce cytokines. And what I want to point out here is like you see that the number of cytokine producing cells is higher than detected by the AIM assay. So 50% of the cells of the antigen-specific CD8 T cells already induce cytokines after the first dose. This rises to 70% after the second dose, and maintains like more than half of the donors are able to produce cytokine responses 7 months out after infection. So that was very encouraging, because that means we can not only detect antigen-specific cells, but they are also functional and do acquire multifunctional responses after first and second dose of vaccination. So in summary, what I've just been showing you, this is all of the results. So we've seen a response or induction of antibodies and CD4 T cells in 100% of the people after second dose, and this is maintained in almost everybody, depending on the parameter you're measuring, 7 months out. So you can see we walked through like 100% of the donors still have detectable antibodies. 88% of them are highly functioning. 97% are responding with CD4 T cell response 7 months out. And overall, by either of the two assays measured, we still see detectable CD8 T cell responses 7 months out after infection, after vaccination. Apologies. So I pointed out in the beginning that what we have been analyzing is not the vaccine dose that is typically given right now, but the quarter of the dose, 25 micrograms. So we, of course, were interested in how does this compare to natural infection. So we picked samples that we knew have had an episode of COVID-19, and we collected samples, longitudinal samples for them, like 6 to 7 months out after infection. So this is what we have been using to compare with response induced by the low dose of the vaccine 7 months after vaccination. And you can see here, for any of the antibody parameters, we did not see any response between the low dose of the vaccine or natural infection. The same is true if you look at CD4 T cell responses or CD8 T cell responses, which are in the right part of the slide. It doesn't matter if you look at AIM-specific cells or if you look at cytokine-producing cells. Neither CD4 nor CD8, there is no difference between these spike-specific responses induced by like the antigen-specific response or by the low dose of the vaccine. So that was encouraging, because that means even a low dose is able to give you like responses comparable to like people that had natural infection. But of course, the question that everybody was asking was, how does this compare to the 100 microgram dose? Because like this is what we have been giving everybody. So this is what we looked next. And again, I start with the antibody responses here. So the 25 microgram dose, what we have been studying, is shown in the gray balls, and the 100 microgram dose is shown in the green balls here. And you can see all of antibody parameters that have been measured are higher in the 100 microgram dose. So this is not, we are not the first ones to show that. That has, of course, also been shown by like Moderna, and these are the two citations that I wanted to point out here. So antibodies response by 100 microgram dose are higher than 25. And that is ultimately probably also why the 100 microgram dose has been used to like going forward for this vaccination. What about T cells? So if you look again at CD4 T cells on the left side of the panel, and CD8 T cells on the right side of the panel, you can see that similar to what we have been seeing for antibodies, also CD4 T cell responses are significantly stronger induced with the 100 microgram dose, as shown on the left by either the AIM assay or the ICS assay. But interestingly, we didn't see any difference in the induction of CD8 T cell responses, as is shown here on the right side. So it doesn't matter if you look at the AIM or the ICS, you see the exact same magnitude or multifunctionality induced by the high or the low dose. So to summarize this part, we have been shown that 25 microgram of this mRNA vaccination induces spike-specific IgG, RBD IgG, and neutralizing antibodies, as well as CD4 and CD8 T cell responses that are very comparable to natural infection. We did also show that 25 microgram of this vaccination induces lower spike-specific IgG and neutralizing antibodies, as well as CD4 T cell responses compared to the 100 microgram dose. This is the one that's given right now. We also shown that this low dose of vaccine induces comparable magnitude of CD8 T cell responses compared to the 100 micrograms. So that was like surprising, because that doesn't make any difference if you have the low or the high dose. But if you look in close detail, the frequency of positive donors was a little bit higher in the higher dose group. So this was very encouraging because we were able to see after the low dose a long-lasting induction of immune response 7 months out. And so we could answer the first question we had, how long does T-cell memory last in these vaccines? And we showed you with all of the different parameters measured that also in these donors, majority of donors, we can readily detect responses 7 months out after vaccination. Even these donors have only received a low dose of the vaccine. So now we were interested in answering the second big question we had, and this is, does pre-existing T-cell immunity influence the outcome of vaccination? And this was the perfect cohort we were having because, as I said, it's important to have a pre-bleed. That means like you have a sample available before these people have gotten the vaccine. And that's important because like only if you have a sample that does not, that has been collected before vaccination, you were able to determine if the donors have or do not have pre-existing T-cell memory. And we did this analysis on our cohort on the day 1 sample, which is basically right before they got the first dose. And we saw that our donors almost fall half and half. So half of the donors do have spike-specific CD4 T-cell memory, half of the donors did have not. So that gave us the perfect opportunity to now divide our groups and see if there's any difference in people that have pre-existing T-cell response, which is shown here in the blue balls, or the ones that do not have that. And the middle graph just shows you that all of these pre-existing cells do actually come from the memory, T-cell memory response, just as a confirmation that what we see is truly derived from the memory compartment. The first question we asked, shown here in the very right part, is, does this CD4 T-cell response that is induced by the vaccine has any difference in people that have the pre-existing immunity or do not have the pre-existing immunity? And you can see here at all time points studied, there is a higher response frequency of antigen spike-specific cells in people that have pre-existing immunity. So that means not only can they induce a faster response, but also a stronger response. And that was true at all time points. We measured all the way 7 months after vaccination. We see that there is a stronger response in people that had like 7 months ago pre-existing immunity. What about the other parameters? So I told you that we are also interested about the functionality, multifunctionality of the T-cell response. And again, we divided the donors into the blue group, which is half pre-existing immunity, and the one that did not. And we saw that the ones that do have pre-existing immunity were able to induce faster and a more higher frequency of multifunctional cells after the first dose, but also after the second dose of vaccination. So this also looks like donors that have pre-existing immunity are able to induce faster and stronger multifunctional T cells compared to people that did not have pre-existing immunity. What about the circulating T follicular helper cells? I told you that these are important because you wanted to have this subset because these are the CD4 T cells that help generate more robust antibody responses. And again, here we saw that in the early time points, which is like the second after the first dose, the second time point, which is after the first dose of vaccination, significantly higher induction of circulating T follicular helper responses compared to the people that did not have pre-existing immunity. That difference went away if you looked further out after vaccination, but clearly after the first dose, you have an advantage if you have pre-existing immunity. So I told you that why we measure TFH cells is because like they are helping antibodies to develop higher, stronger multifunctional antibody responses. So we asked next, if we divide our groups in pre-existing, yes or no, do we see a different antibody responses? And the answer is yes. Also, like in the antibody response, so if you look at spike-specific IgG titers, you do see a stronger antibody response in people that did have pre-existing immunity compared to the ones that have not. Similar to the T follicular helper cells, also like this response is most notable in the after the first dose, and then is going away the difference after the second dose or in long-term memory. So similar things we have been seen if we not look the entire spike-specific IgG, but the RBD-specific IgG titers, where also we saw like a strong induction after the first dose that is then like fading out the difference in the other time points. Very interestingly, though, I told you that it's important to not only always measure the level of antibodies, but also the functionality. And very interestingly and importantly, if you look at the neutralizing capacity of these antibodies, these are significantly higher 7 months after vaccination if you do see people that have had pre-existing memory 7 months out. So this is important. So if you had pre-existing CD4 T cell responses, you have a higher neutralizing capacity 7 months after vaccination. So basically, we can conclude from this part and answer this question that individuals with pre-existing spike-specific CD4 T cells generate early multifunctional CD4 T cell response, a more robust follicular helper T cell response, and faster antibody response after a single immunization. So they truly have an advantage compared to people that do not have pre-existing immunity. And with this, I like to finish my talk. So I hope I convinced you that we could find answers after the 2 questions we had. This is the team at the Jolla Institute that has been working together like for the last 18 months. And I have been acknowledging people that have been doing the different parts throughout the presentation. And with that, I like to stop and please feel free to email any questions if you have any. Thank you so much.